All right, here's our first video lesson that I'm hoping will help clarify material from our slideshow and highlight the things that I think are really important. And as we go through nucleic acids, this is the last of our biological molecules that we're going to look at. And again, we want to keep the idea that chemistry drives the biology. And so as we look at the DNA and the RNA, we want to understand what the chemistry is and how that specific chemistry is important to its biological function. So hopefully this helps give you some highlights, and then Mr. Allen will take you through the rest of the slideshow and you can ask questions together as a class. Through this lens of chemistry driving the biology, the chemistry of nucleic acids is all about nucleotides. The nucleotides are the individual building blocks here, and they will determine the characteristics of that molecule, whether we're talking about DNA or RNA. Now, the nucleotides are very similar, but they have slight chemical differences, and these nucleotides are repeating subunits through a nucleic acid. So similar to the carbohydrates and the proteins that we looked at, the nucleotide is a polymer in that it's made up of many smaller building blocks or many monomers. So we're going to look at that nucleotide and see how it is unique. Nucleic acids come in two different forms. There's DNA and RNA. DNA, as you know, determines the heredity of the cell, and you looked at this in grade 11 biology. RNA, as you may not be aware, is specifically involved in making proteins for the cell. And when we get to our genetics unit, we're going to look at this flow of information. That information goes from the DNA, which is unique to you, and that genetic code, that chemistry, will ultimately determine RNAs that get made, and the RNAs are used in the cell to actually make the proteins, and the proteins are what do everything. And in our genetics unit, we're going to focus in on that relationship and that pathway from DNA to RNA, RNA to proteins. And so we need to look at the specific nucleic acid, the nucleotide structure of both of these. So here's the individual structure for all nucleic acids. They all are made up of what's called a nucleotide. This is our individual building block. And if you look at this picture, it kind of looks like a house maybe that we have here in the front. We got a garage off to the side, that's our base. And then we got this phosphate, this pool in the backyard. I saw one video that described it that way. But these nucleotides are all unique, and they are different between DNA and RNA. We'll look at the sugar is actually unique. What type of sugar do we have here? What are the bases? Because there's 4 different types of bases. And then what is the bonding that holds those together between DNA and RNA? And those all play a role in the chemistry. If we look at DNA and RNA and we look at the sugar itself in this base, it's a 5-carbon ribose sugar, but we notice that DNA is deoxyribose. And the deoxyribose, just to highlight here, we've removed a hydroxyl group from the RNA, and now it's deoxyribose, okay? And that makes a bit of a difference. Having that hydroxyl removed makes the DNA less reactive, which is important because we want our DNA to last a lifetime. We don't want it to break down or get broken apart, and so it's deoxyribose. A small change in the chemistry here has just changed some of the integrity of this nucleotide in lasting longer. Beyond that sugar, you'll see that DNA and RNA have some different base pairs. They all have adenine, guanine, cytosine, but you'll see here that the last one, the 4th one, DNA has thymine and RNA has uracil. And when you're looking at those, I'm happy for you just to remember it's an A, a G, a C, and a T, or an A, a G, a C, and a U. But those bases are a bit different, and thymine is actually more stable. It doesn't break down as readily as uracil, and so again, for DNA structurally to hold the integrity of the information it has for the lifetime of that cell, we need a more stable base. Uracil can actually break down a little bit more, but for a messenger, that's fine. Uranil is kind of like the off-brand version of thymine is how sometimes I think of it. Finally, we'll see that the bonding is different between DNA and RNA. The bonding pairs in DNA are adenine and thymine, guanine and cytosine. And if you put these in alphabetical order, right, the outside pairs bond together and the inside pairs bond together. But the chemistry between an adenine and a thymine will make it so that they fit together. The puzzle pieces fit together, and hydrogen bonds will naturally hold those together. And C and G, again, the chemistry will hold those together. So it helps ensure the integrity of our code and we have the right matching pairs, that those bases will bond together. In RNA, we have similar kind of structure, but again, we have a U, right, instead of a T. But again, if we're making proteins, the DNA and RNA would match up, and we'd have these kind of base pairs. The adenine and the uracil would go together, and again, guanine and cytosine would go together. When you put DNA structure together, you see that it's double bonded. It's double bonded, so it holds together. It's more protected. Again, when you think about the genetic code and we got to protect it for a lifetime, this is really important. RNA is single-stranded, so it's a little bit vulnerable to break down, but as a messenger, that's okay. DNA has some unique characteristics, and we're going to come back to this in our genetics unit, but we want to highlight for you that the direction here, this anti-parallel characteristic, is important. Things go from a 5' end to a 3' end on one side, and it's anti-parallel in that on the opposite side, we go from a 5' end to the 3' end. And the 5 and 3 are just related to the sugars in that ribose group. Remember how chemists number the sugars. So we see that anti-parallel nature, which is important to distinguish. We also need to distinguish the bonds that hold things together. We have these phosphodiester bonds, and those are common in DNA and RNA. But in the double-stranded DNA, we see the hydrogen bonds, again, the A's and T's, G's and C's, that hold this together and help protect this molecule. That chemistry is really important. Ultimately, the order that these bases come in give the genetic code, from whether it's your goldfish in your room, your dog, the plant outside your garden, or you. The order of these A's, C's, G's, and T's determine who you are. That chemistry is crucial to the biology and ultimately the code for the proteins that make up who you are. The chemistry drives the biology. Finally, the last thing we want to look at is another role for RNA, where we look at microRNAs. And microRNAs we've known about them for about the last 20 plus years, but we're starting to understand more how this group of microRNAs plays a key role in regulating gene expression. Example, what that's saying is it helps control what proteins are made and how many are made. And this is crucial to the health of individuals. It's been noted in a number of diseases, including cancers, heart disease, and neurological diseases. These microRNAs are only 22 nucleotides in length, so they're extremely small. But appreciate how the chemistry, the order of those nucleotides, is very significant in determining the ultimate biology, what proteins are made and how many are made. You're going to watch a TED Talk that looks at some of this early diagnosis and the impact that these microRNAs can have, and understanding them, how we could maybe diagnose diseases at a better rate. If you don't have a biochemistry topic at this point, this is something you might want to look into a little bit further, because these microRNAs give us an insight into the health of the cell. I hope this short video helps highlight the information that I want you to take away from this lesson. Keep that framework of chemistry drives the biology first and foremost. Ultimately, when we get to our unit test, we're going to be looking at, can you support the case that the chemistry drives the biology? And could you use nucleic acids as an example of how the specific chemistry, whether it be DNA or RNA, how that specific chemistry impacts the biology? Ultimately, the genetic code, the A's, C's, G's, and T's that make up our DNA, they impact the proteins that make up you and I and every other living thing. And so that chemistry is crucial to the biology. Good luck.